Hey, I just wanted to quickly plug that if you guys want any tutoring, some new slots have opened. If you look in the description, there's a link to my physics and maths tutor profile in which I do tutoring on there. Hello, everybody. Welcome back to another episode of the A-Level Cookbook. Today, we're going to be doing the OCR A-Level Biology A, June 2019, Paper 3. So let's get started. So sago pondweed is an underwater plant that grows in many regions of the world, and here's a transmission electron micrograph of this pondweed cell. So here we have A here, and we've got B here, and then C and D. It says identify the cellular components shown at A and B. So with A, it's a big hole, an empty space. So here, it is a vacuole, right? Whereas with B, if we have a look, this is B within this bit here. So we've got a nice big black dark spot here within this. That must be the bit within the nucleus, so nucleolus. There. The real size of the line between C and D is this. Calculate the magnification that was used to produce that figure. Give your answer to significant figures. All you've got to do is you've got to do I equals AM, where image equals actual times magnification. Magnification equals image over actual, right? You need to make sure the units are consistent. And what you would do is you would just measure this line with your ruler, convert the units to meters here, do I over A, and then you can work it out like that. I can't do that here because I don't have a ruler and I've tried and it don't work. So in this case, it's just going to be times 14,000. There you go. Figure 1.2 shows a student's drawing of another sago pondweed cell which is observed under a microscope, and they used a sharp pencil but did not label it. Describe two other ways in this that can be improved. So here, there's shading. There should not be shading. So no shading. As well as that, there is no scale or magnification. Immediately I can see that. Other things you could talk about as well is that there's no title either. And obviously the question's already told you it's not been labeled. So now it says the student stained a sago pondweed sample to improve the contrast between cellular components and viewed under a microscope, and they used the following method. So they put forceps to place it on a glass slide. They used a pipette to place 2 drops of stain in the center of the sample. They lowered a cover slip onto the sample, ensuring that the cover slip is parallel with the slide as it's lowered. Describe 2 improvements that they could use. So here, the issue is imagine you have a sample and you've got this and you put the stain in the middle. It's going to spread out like this, right? But this bit's going to be more concentrated. It's going to spread out like that. So it's always better to put the stain on the an edge of a sample, not in the middle, because then when you're lowering the slide onto it, you're pushing that stain this way outwards. It's spreading out nicely. Because if you're going to do it like this centrally, then it's going to be more concentrated towards the middle as you're putting that slide on. You're leaning the slide over, if that makes sense, lowering it on. Here, what you would do is you'd place stain at edge, not center. What else could we do then? So it says here, carefully lower slip onto sample, ensuring that the cover slip is parallel with the slide as it's lowered. What they're doing is they're literally just plonking it down like that. You shouldn't really do that. It's better to sort of do it at an angle and lower it down, right? Or if you can, use a mounted needle. So in that case, you would lower the slide at an angle. There you go. Other things you could do as well is I've seen as well, you can say using more than one stain because you can get even more contrast if you really stain different bits and bobs that stain specifically. Those are really it. And then I guess you could remove the excess stain as well by using like blotting paper. But those are the ones that I see straight off the bat. Sago pondweed has evolved many adaptations to its aquatic environment. 3 of these are described below. Explain the advantage of each one. So no waxy cuticle. We need to think about why that's a helpful thing here. So we need to think about why plants would have a waxy cuticle to begin with. The reason is because it reduces the amount of water loss through the surface of the leaf through transpiration, right? Because plants don't really want to lose too much through transpiration because, they will have less photosynthesis and all that kind of stuff. Transpiration, by the way, is just the loss of water vapor from the leaf as it evaporates and diffuses out, right? But that then drives cohesion and tension and all that jazz. Here, why they have no waxy cuticle? They live in the waters. It's not really a problem, is it? So then, if they're wasting energy and resources and making waxy cuticle and they don't need it, then that's a bit just not a good idea, right? So there's, water loss is not a problem. There you go. So then it has stem tissue that contains air spaces. Why is that a useful thing? Here, stem tissue that contains air spaces an aquatic environment, right? If you are a plant living in the water, you don't kind of want to be like that because you still want to get as much of that sunlight coming from above to photosynthesize. If there's air spaces within it, air floats in water. So what it does is it'll make it become buoyant and help it to, be closer to the surface or float, if that makes sense. The reason that fish can float, for example, is that they have air in them that's buoyant and they're less dense than water as well. If you put there's what's those things? Those circular donut things that you swim with, they have air in them to help you float. So here as well, it's to give them buoyancy or to allow them to float. To allow them to float, which means they can be closer to the surface of water, which, can let them be closer to light for photosynthesis, or it could be that they're, for gas exchange as well, because, towards the deeper parts of the ocean, you're not going to have as much, as much easily provided CO2 or O2 and so forth. Now here it says a thin, flexible stem. Well, if we have a think about it, we're living in water, right? If I have this rigid, rocky stem here, right, then it's just, I'm fixed, I can't move. But if I have a thin but not very flexible stem, it's rigid, and there's currents moving and fish banging into me and all that kind of stuff, I'm going to snap. So if it's thin and flexible, it means you're not going to be as lightly damaged, really. So less likely to be damaged by water currents, is what I'm thinking. It's all linking back to, why would this be useful in a thing that's living underwater? That's what I'm thinking. So figure 2.1 shows a naked mole rat. Fine. The naked mole rat is a mammal, however, it has several features that are unusual for mammals. So they regulate the temperature in a way that's different from most mammals. Some of the thermoregulation in naked mole rats is as follows. So they live in complex underground tunnel systems which have a stable temperature of this. However, sometimes the environmental temperature can increase to decreasing outside this range. In experiments that examine environmental temperature changes, the core body of naked mole rats remains close to that of the environmental temperature. When the tunnel temperature increases, they often move to cooler parts, and when the tunnel temperature decreases, they often lie together in large groups. Outline 2 ways in which this is different to thermoregulation in most mammals. So here, already, when we are, for example, like humans are too warm, we sweat, we chill out with the respiration, we get all like, oh, I'm going to sit down and just sit and stuff, right? Whereas here, they seem to be moving actively to cooler parts. As well as that, when our temperature is too cold, it seems according to this, they go and lie together in large groups. Whereas mammals will shiver, right? So here, there seems to be more to do with them doing things, as in more behavioral responses rather than physiological. So immediately, one of them is that there's more behavioral responses than physiological. You can't assume that they're shivering. You have to work on the text, what it's told you. Physiology is just means like processes that we do, things like chemical reactions, this thing moving to do this, like the automatic-y, processy stuff. So like you could even just say they hold it together while they don't shiver, for example, whereas other mammals would like sweat. What else is different then? So, as well as that, it's told you in the question that it has a stable temperature of 30 to 32, but sometimes it decreases outside this range. And in this question, it's told you as well that this can remain close to that of the environmental temperatures. It's going out of this range, and the mole rats also have the temperature as well as outside of that range. Whereas humans, we need to keep ourselves at about 37 and maybe 37.5 degrees, right? So there's not a narrow range like we have, for example. So they have so the temperature is not maintained in as narrow of a range compared to mammals. You can't just say naked mole rat temperature matches environment. That doesn't mean anything. It's comparing it to mammals. Mammals are always oscillating between the two here, but for some reason, these may also be oscillating between the two, but it might just kind of like match like this rather than kind of stay around the middle, if that makes sense. We don't really do that. So in humans, when core body temperature falls below 35 degrees C, positive feedback causes this decrease to continue, and this is known as hypothermia. Explain how this could accelerate the process of hypothermia. Negative feedback is where a factor changes and you want to bring it back to normal. So here it's too high and then it goes back to normal, but maybe sometimes you overshoot, so we bring it back up and down and up and down, right? Now, for example, in insulin and glucose, insulin lowers your blood glucose, whereas glucagon makes it go higher. So if your glucose is too high, insulin brings it down, but then obviously insulin overshoots, then you have glucagon to bring it back up, and then insulin brings it down, and so forth, right? Imagine if you just had to depend on insulin to bring it down, and you had to just kind of wait until you ate something to come back up. There's nowhere near as much control. And that's just a side point as well. With negative feedback, we have things usually that bring it down and up. Because imagine if you drove a car and you had no brakes, right? The only way to slow down is to just let go of the accelerator and eventually just wait to slow down. Right? Whereas if you have brakes, you can slow down, speed up, slow down, speed up, right? Now here, the question's told you that a positive feedback loop causes a decrease in temperature to continue. So you got a low temperature, and then for some reason, positive feedback makes it go lower. Positive feedback is where that change is amplified, right? So if I have a fall in my temperature, rather than going back to normal, what happens is it actually goes worse. So that's what it is. Homeostasis is the maintenance of an internal environment, and that's nearly always actually, negative feedback. Positive feedback is where you're going further away from that. So here, we're going to say what positive feedback is, right? So positive feedback exaggerates a change away from the normal. I think it's always helpful to say this either towards or away from the normal, or optimum if you want, because it really makes it clear that you understand what you're saying, from the normal, all right? But that's not us done though, because we need to figure out about how this could accelerate the process of hypothermia. So we need to think about what might be going on to make this even worse. So here we have low temperature, and now for some reason we're going even further. We need to think about why. So how do mammals get their temperature to go up? What do? We shiver, right? But we also respire. So respiration as well releases energy in the form of heat, right? As well as the fact that it's put into ATP and so forth. So if we have a low temperature, we need to think about the ways that lead to respiration slowing down. So low temperature means we're going to have low kinetic energy because it's respiration and all of our metabolic reactions really are enzyme mediated. So if we have less kinetic energy, it means that we're going to have decreased respiration, which means less heat's generated. And as a result, the body temperature drops even further. And there you go. So mammals, including naked mole rats, have temperature receptors that play a role in thermoregulation, and here's 4 statements about mammalian temperature receptors. Write either true or false in the empty boxes to indicate whether the statements are true or false. So peripheral temperature receptors detect the temperature of internal organs. Peripheral means outermost parts like your skin and further outy bits. Internal organs are in the center, they're central, so that's false immediately, right? Receptors in the hypothalamus detect core body temperature. That is indeed true, they do. The thermoreceptors there do. As well as that, body temperature is detected by receptors. Blood temperature is detected by receptors in the hypothalamus. Well, yes, that would also be the case because they're detecting the temperature of the core using the blood, so that's true. Temperature receptors send impulses to the medulla oblongata, which regulates body temperature. That's incorrect. So receptors detect a change. They detect a stimulus, a change in the external environment. Here, the temperature is changing, so we need to send an impulse to a central processing thing of some sort that detects and mucks about with this change, right? From these temperature receptors in other parts, right? So they send it through a sensory neuron to somewhere in the central nervous system, which then sends impulses along to an effector to make a change happen. Now, in this case, it's the hypothalamus, not the medulla oblongata. Medulla oblongata regulates things, for example, like breathing and your heart rate. So immediately that one's out as well. It's this. The temperature receptors here, remember that you can have receptors in the hypothalamus itself at the core and blood stuff, but temperature receptors are the ones that are more distal as peripheral ones on the outside of your skin and so forth. So another unusual characteristic is their tolerance of pain. Acid causes pain responses in most mammals. Naked mole rats are tolerant of the pain caused by acid. This tolerance can be explained by the type of pain receptor found in naked mole rats. So here's a representation of the ion channels and the pain receptors of naked mole rats and other mammals. Let's have a look, shall we? So here we have a cell membrane. We've got an acid-sensing Na+ ion channel that opens in the presence of H+, right? More H+, more acidic. Here you have a voltage-gated ion channel. This is prevented by the opening if you have H+. So if you have more H+, this ain't opening, right? But this will open if you have more H+. So if you have a lower pH, this will open, and a lower pH, because lower pH removes more H+, more acidic, not open, right? Whereas here, acid sensing, this opens in the presence, so lower pH opens, and then here, this channel is not affected by H+ ions, so it may be open depending on this. So it says, use this information to explain why naked mole rats do not feel pain when exposed to acid. So here, we've just established that this will open if there's H+ ions, right? So that means if the decreased pH, more acidic, these will open. So then remember that sodium ions moving into a neuron will make it more positive. If they move in, you get a generator potential. If you hit the threshold, if you have the right generator potential, then V-gated Na+ ion channels will open it. Bang, there's your action potential. Then they close, right? And that stops increasing. The only way to get it to come back down to repolarize is the potassium ions moving out, and then like this and so forth, right? Here, we're able to get that generator potential possibly. This is what we have to work with here. These acid-sensing Na+ ion channels open so they can move in. However, the voltage-gated ones, these only open when a certain voltage is met, as in the threshold, aren't open if you have more of this. So if there's a lower pH, as in it's more acidic, these cannot open even if you hit the voltage, which means you don't get an action potential. If you have no action potential, you're not sending impulses along the sensory neuron to your central nervous system, you're not processing the pain. Your central nervous system hinges on action potential there, action potential no. And your brain can only sense the things if the action potentials are getting to it. So here, in this case, is that the lower pH means that V-gated, and you can't just say sodium, you have to say sodium ion, Na+ ion channels cannot open. This can still get you a generator potential, remember. It doesn't mean you can't, it's just this can't actually hit the threshold. So they cannot open even if threshold met. Therefore, there's no action potential. There's no impulse along sensory neuron. There you go. Explain how a pain receptor is an example of a transducer. So a transducer is something that produces an electrical impulse based on some sort of stimulus, right? So for example, here it would be there's a chemical stimulus. So it converts a chemical stimulus into an electrical impulse. It's transduction is where you're changing it from one form to another. So for example, pressure stimulus, for example, in your skin, when you're pushing an apaxinian corpuscle, turns it into electrical impulse as well, electrical energy. That's transduction. Right, so figure 2.3 shows the relationship between body mass and lifespan. Here we have our naked raw mat, and you can see there's a roughly positive correlation between the two of them. Fine, whatevs. Here, how would you look at that? Describe the relationship between the two of them. There is a positive correlation. Wow. Now it says, what conclusion can you draw about the lifespan of naked mole rats to other animals? So here, can you see how this is pretty high, but it's not fitting the majority of the results? Like you'd expect to be following this line. If we drew a line of best fit through here, like this, right? It would kind of be like, almost like, whoop, like that, which is a bit odd. It's outlying, it's unexpected, it's weird. So what we need to do is we need to turn that into words and actually use it in the context of the graph. So if we were following its lifespan here, we would expect it to be somewhere around here, right? If we're following its mass, sorry. But we can see that its lifespan is higher than what most of the other ones are. So its lifespan is higher than expected for its mass. So lifespan higher than expected for its mass. There. Naked mole rats could survive without oxygen for up to 18 minutes. This is several times longer than other mammals of a similar size. The following information might help to explain why they can. So in normal glycolysis, the enzymes needed to convert glucose to triphosphate may be inhibited by lactate. Naked mole rats can use fructose as a respiratory substrate. Fructose is converted to triphosphate, and triphosphate can then enter the glycolysis pathway. Suggest why the use of fructose allows it to survive without oxygen for a long time. So remember, let's have just a quick look at this, what's going on, right? Glycolysis, the entire point is that you have glucose, and then eventually you get to pyruvate, right? Now, if you have no oxygen, what you're going to end up doing is you're going to make lactate from this through anaerobic respiration, lactate fermentation, because we're not doing the link reaction, right? And the questions told you that the enzymes needed to convert this inhibit the glucose being converted to pyruvate. However, here it's told you they can use fructose, which is converted to triphosphate. So glucose is, remember, converted into hexose bisphosphate by adding two ATPs on, phosphates from ATPs on, and then you get TP, right? And then eventually that gets oxidized, you get pyruvate. Now, the questions told you that they can use fructose as this. It's converted to triphosphate. So we have fructose, which ends up being TP, and then bang, there you go, right? And then it's able to do the rest. So then it says, suggest why the use of fructose allows to do that. Well, it's just been established there. You're able to continue anaerobic respiration because the glucose to TP is not essential here because they can use the fructose instead, right? But that still doesn't explain how that's helpful. It's because when you are converting TP to pyruvate, right, you're oxidizing it, but you're also making ATP in the process. So ATP is produced there. And that's what allows them to survive because they can still make ATP. There you go. So just one other aspect of the physiology of naked mole rats explains how they're able to survive without oxygen for a long time. If we have a look at the previous questions, remember that they said in the question their temperature matches the environment, which is low as 30 to 32 degrees. And when it's too warm, they move to cooler areas. And when it's too cold, they lie together. They're not using as much energy heating themselves up by doing more respiration, right? They're just physically moving and just having to lie down. That's it. So here, there's two things you could say. Either one, they have a lower body temperature by default because they, again, can go as around, 30 to 32 degrees. But also you could say, or they use less energy on thermoregulation. So they don't have to, use all of their resources and materials up and, glucose and all that jazz to do that. Easy peasy. There you go. Ice fish live in very cold water. They have biological molecules that allow them to tolerate cold temperatures. Adaptations could be three groups. Which category is best, to describe these? So the three ways you can have adaptations are behavioral, physiological, and anatomical. So behavioral means a way that an organism is behaving, things it does. For example, like before with the mole rat moving to certain areas, or like a possum playing dead before it's been killed by a thing, right? Physiological is processes that happen in your body, like metabolism, respiration, right? Shivering, things like that. Anatomical is another word for structure. So structural or anatomical is all to do with how things are built, structure. So for example, like whales and how they have lots of blubber, or like elephants and their big ass ears, right? Here, it's told you in the question that we have biological molecules that allow them to tolerate cold temperatures. So if they are able to tolerate cold temperatures because of molecules, then there are processes happening in their body to let them do that. So it's got to be physiological. There you go. One example of a cold-tolerant molecule present in ice fish is a modified version of this protease enzyme called trypsin. Figure 3 shows how trypsin is converted from a molecule called trypsinogen. This conversion happens in the lumen of the small intestine. So we have trypsinogen with enterokinase, and then we have trypsin with a CA2 plus binding site, and this is in the wall of your intestines. Fine. So it says, state two conclusions that can be drawn from this figure about the roles of the molecules and how they affect trypsin's functions. So clearly, one thing is that trypsinogen comes here, binds to this, and then bang, all of a sudden trypsin comes out. So it must be that enterokinase converts trypsinogen to trypsin. That's what they're asking what that diagram shows, essentially, right? But as well as that, now we have a CA2 plus binding site. Where else have we seen CA2 plus binding to things? For example, in muscles, we could see a tropomyosin troponin. It binds to those and then sort of moves out the way. So if there's a binding site here, then it must mean that it's acting as a cofactor. So CA2 plus can act as a cofactor. So coenzymes, remember, are things that basically just transfer one thing to another, right? So like, for example, NADP, when it's reduced, transfers electrons and protons. Whereas cofactors are things that bind to an enzyme, and what they do is they'll help that enzyme do its job. So if the cofactor binds to it, it'll make the active site more complementary in shape to the thing, for example, and then lets it do that. So here, they've told you in the question that we have a protease enzyme trypsin, right? And there's a CO2 binding site here as well. So there's further evidence to say why this would act as a cofactor instead, because it'd bind to this and it's an enzyme and make it work better. So a group of students investigate the effect of temperature on the activity of two forms of trypsin, human trypsin and ice fish. Part of their method is here. So they get 10 centimeters cubed of 5% trypsin solution, measure enzyme activity at 10, 20, 30, 40, 50 degrees, each trial at the same pH, repeat them 5 times at each temperature, measure the enzyme activity recording the area of gelatin on a photographic film that's broken down over a set time period, and then they calculate the enzyme at the rate at each temperature. So there's a lot going on here. Write down the independent and the dependence, right? So the independent is our temperature, and as well as our dependent is how long gelatin takes to break down, right? And it's timed. So here, when we're looking at improvements and explanations for experiments, what you should really do, and if we're looking for increasing the validity, which is basically, can this be repeated? Is this answering the original question? Can this be reproduced? Is this applicable to every single thing in the same scenario? Always look at what screws up the dependent and whether the independent's been appropriately done as well. Is this an appropriate thing to measure for the question they're looking at? And is there anything screwing this up? So if we look at the validity here, they're looking at how long this gelatin, takes to break down. And they're saying, okay, this must be to do with the activity of trypsin. Is this an appropriate thing? Maybe. I mean, we don't know, right? But if we have a look, if we want this experiment to be valid, we need to make sure that it's doing the right thing. We want to make sure as well that this temperature is the only thing that's messing around with this. If we have a look at each thing step by step, we want to look at controlling whatever screws this up. So pH, which has already been mentioned, right? Another factor we could talk about, for example, is, because we're using an enzyme, we need to think about what's going to screw up the enzyme. So pH has already been dealt with, but we have absolutely no idea about the concentration of the gelatin. So one thing you could do is control the concentration of the gelatin that's been used within the area, because otherwise this could increase or decrease the rate of the breakdown, because it's an enzyme we're dealing with. Right? Other things we can also do is, for example, let's say I get like a very fast one here at 40. I don't know if it was 36, 37, 38, 39. So what I could do is I could use smaller intervals. If I use smaller intervals, then I can identify a more accurate, so closer to the real value, temperature that's optimum. Right? Because if the optimum is like, I don't know, 36, I won't know because I only see 30 and 40. I can't just guess. I have to go and measure it. As well as that, you should really run a negative control, as in you have gelatin without trypsin in. The reason is not because to allow comparison, because you're going to compare all of these. It is to make sure that the trypsin in it is breaking down the gelatin and not the heat. Because if you do this with a no trypsin in, and you see that the temperature is going up and it's breaking down, then the heat is also breaking it. So generally speaking, if you're going to write about control, please do not just say to allow comparison, because that's what we're doing with all of these. The idea of a control is to make sure that your independent is the only thing that's changing the dependent. If you're doing it without the trypsin, because we're looking at temperature screwing up trypsin, right? If I take the trypsin out and temperature is doing this to this, right, without the trypsin, then that's a bit odd. So now it says suggest appropriate units to represent the rate of enzyme activity in this investigation. So again, because we know our nicely dependent variable is, we know it's going to be the rate at which, or how long, the rate, I should really say, of an area of gelatin being broken down. So it could be in centimeters squared, but it's in rate, so per minute, right? So centimeters squared, minute to the -1, for example, or second. Depends on how quick it is. So students recorded the temperatures and produced the fastest reaction rate in each of these 5 replicates, and this table 3 shows these results. Here's our replicates. This is the temperature that produced the fastest one. So these are all the fastest. These are the fastest. They've got the mean, median, and mode, whatever, right? So here we can see in humans, the fastest tends to be 40, 30, 40, 40, but there's a weird 10 here for some reason. Whereas in the ice fish tends to do best at like 30, 30, 20, 20, like around that sort of thing, right? So the mean in the human trypsin is higher to get the fastest rate, the temperature, than it is in the ice fish. Cool. So then it says, I think the mean is a more accurate measure than the median or mode for these results. So it's a bit of a 50-50 because it says evaluate, so we need to talk about supportive and contradicting, right? So if I look at the supportive statements, if we look at the other two, the mean here is 32, that's 22. If you look at the mode, it's 40, but then here there's 2 modes. It's not really good to have 2 modes. You can't really compare them. That's not very helpful. And as well as that, the median is just the middle value, which for median, right? In fact, the middle value is not, it's all right. But like, for example, if I have a gigantic range from 0 to 10,000, and my middle number tends to be 50, it doesn't really encompass all of that. Whereas the mean might be really, really big. It might be in the hundreds, if that makes sense. So I guess a positive here is, well, first of all, there's 2 modes in there. So there's 2 modes in ice fish. So it's harder to compare. And I think that's the stronger point, really, because the median isn't too outlandish here, to be honest. But in that example, it is. Now, the contradictory statements are the problems with this. So what's the problem with using the mean versus the median or mode? So let's look. The mean here right? You see how there's that weird number there? That's going to drag down the mean. If I measure like 100 grams for a chocolate bar, 120 grams, and then for some reason I get 10,000, right? My mean is going to be 100 plus 120 plus 10,000 divided by 3. My mean is 3406 grams, or 7, sorry. Right, that's implying that like most average chocolate bars in this sample I have is 3,400 grams, which is definitely not, because they're usually 100. But if I then measured, for example, 300 more of them and they were 100 grams, right? Okay, so now my sample is 300 100 grams plus another 100 grams, so 120, and another 120, and then a 10,000 grams, right? That's 301, 2, 3, 4. That's 304 measurements. My mean is now 132 in this new one. Can you see how that's way closer to what the middle or the most commonly seen thing is, right? Here, they've only got 5 replicates, and that mean has dragged everything down here. In the same way, my mean got dragged up by these mini, like this tiny sample here. So in that case, one of the negatives of using this is that the mean includes the 10-degree outlier, which decreases it and makes it less accurate. The mean is less accurate, not the results, the mean, because we want to be close to the real mean value for the mean. This can't give you the close to the real value for the experiment because we're not directly measuring that, if that makes sense, right? As well as that, and if we compare the use of the mean to the mode and the median aren't actually screwed up by those, outliers. Because if we then had a look at my 3 chocolate bar sample, the median number is 120, right? And then if I added a couple more 120s in, then my mode is that, whereas the mean is still screwed over by that 10,000, whereas these two are fine. So it's a balancing act between all of them. So then the students wanted to know if there was a difference between these reaction rates. They performed a statistical test on the mean 5 replicates of the human trypsin and the 5 for the trypsin and the ice fish. What's the best statistical test? Explain why. So they're doing a statistical test on the mean of the 5 replicates, and they're looking for a difference between the reactions. Well, really, we're looking at, remember, reaction rates, they're measuring the means. So we're looking to see if there's a difference between the two means. So we're going to be doing an unpaired t-test. Why? It's because we are looking to see, or comparing, really. We can say we're allowed to compare here. I don't really like comparing, but yeah, we're comparing means of 2 data sets. And the reason it's unpaired is because they're 2 different groups. If I had a group of like people here, right, and I'm measuring their pulse, right, and the mean pulse, and then I'm getting that same people, making them do an exercise, and I'm getting the mean pulse after. This is paired data because it's the same group before and after something's happened, an intervention. So you would do a paired test there. However, here we're not doing that. They're 2 separate groups entirely, so it's an unpaired t-test. So now it says an individual's immune response can change throughout their lifetime. Figure 4.1 shows the immune response by the influenza virus when they were first infected and when they were infected 2 years later by a new mutated strain. The influenza virus has many antigens in which the immune system can respond. Figure 1 shows responses to 4 of these antigens. So let's have a look what's going on. So immune response to influenza virus and then 2 years later. So this is a percentage of the normal response. If you're less, you're having less of an immune response compared to your normal one. If you're above 100, then obviously you have a higher, fatter response. Here, for the antigen here, our normal immune response is 100 for all of them because it's the first time we're seeing it. Fine. No big deal. That's fine. That's expected. Whereas here, if you look at antigen A, can you see how on our second infection, 2 years later, it seems to have been decreased. Our response is actually lower than what it originally was. Whereas here, for B was at the same level as it would have been. That's our, normal response. But here it's higher. So you've had a secondary immune response with more antibodies being quicker and faster in production and higher numbers because of those memory cells, right? C, same as A, seems to be a lower response the second time, and then D is a higher, right? Now it says, explain the differences in the immune response to the influenza virus with their immune response 2 years later. So explain is not just, here's what we see, it's saying why. So if we have a look at, like I just said before, B and D have a larger immune response. And that's just the observation. We need to say why. Because memory, B and T cells produce a stronger, faster, or larger response to antigens B and D. Right? But that doesn't explain A and C. So for A and C, what's happened is that we have a smaller. What might be happening is, because remember, we're looking at the influenza virus getting in our body, and the questions told you that the virus has many antigens, as in it has all 4 of these here. If 2 years later we're getting infected and there's less of a response to A and C, it's probably that this virus doesn't have those antigens anymore or less of them. That's the only reason that would make sense, other than, I guess, us losing memory cells, but that wouldn't make sense because it's only 2 years. And also, it's only been 2 years. So here, it must be that the virus, the influenza, has less A and C antigens now, and we need to think why. Antigens are proteins. Any antigen, it's generally proteins, but it can be like toxins and other stuff. But anything that stimulates an immune response is an antigen. Against generator. That's what antigen means, as in attack generator. So here, Antigens are going to be a protein because it's on a virus, right? So it must have become different as a result of mutations. So here, the virus has got less of these antigens now because of mutations. Because when you change mutations, you're changing the base sequence, right? If you have mutations, you're changing the base sequence. If you change the base sequence, you have different codons, you have different amino acids, you have different mRNA, different translation of it, and then you get different proteins. So that must be the explanation there. The specific immune response involves B and T lymphocytes. There is variation between specific immune responses between individual animals. Variation between immune responses can be influenced by genes and the environment. Using examples, explain how both genes and the environment can cause animals to vary in their specific immune responses. Fine. So the good answers were the ones that kind of went through logically and step by step, talking about how genetic variation had some link with immune system cells. But a lot of people were mixing the term antibody and antigen. Antigen, anti, attack, gen, generator, creator. So it's something that's generating an attack. An antigen is something that stimulates an immune response, as in to attack it or get away from it or like destroy it, right? Antibody is attack, body, thing, attack, thing. Antibodies are made by B cells that attack an antigen, or not attack, but destroy. Don't use the word attack, right? Pathogen, pathos is disease, generator, gen, creator, disease creator. So like something that causes disease. So not all antigens have to be from pathogens. It can be from things that aren't pathogenic normally and just happen to be recognized wrong. So if I put my blood cells, for example, I'm AB positive, I think. If I put that in somebody who's O negative, they don't have AB antigens. So their body sees A and B proteins and is like, I don't recognize this. I'm going to destroy it and creates antibodies and stuff like that. And that in itself is acting as an antigen there. So it's not always a disease-causing thing. So now we need to think about here how genes in the environment causes animals to vary in their immune response. Right? So let's talk about the genes bit first, one by one. So where the question's asking why genes lead to variation in genetics, which then leads to different B cells. In your immune cells, all of them depend on receptors interacting with chemical signals or with the antigen itself, and receptors are proteins. Proteins are made up by, joining of amino acids to get a primary structure, and amino acids come from your genes. Whoa! So genes, let's think about how we can change them up. So we inherit the genes for immune components like, antibodies, for example, and receptors from parents, right? We could have crossing over and recombination, right? Which then means you get different combinations, because that's what gives us genetic diversity of alleles for these components. And then, for example, we could have different variable regions on antibodies. The reason that we are genetically different now and why you're not the same as your siblings, for example, and not identical is because of crossing over recombination and all of that jazz, right? So that's also what's going to lead to this. However, it's talking about our variations in our specific immune responses, which is linked to B and T cells. So we need to think of other possible genetic things that can make your B and T cells behave differently. For example, if I have a normal cell chilling here, and all of a sudden, this normal body cell is getting absolutely battered by an immune response. The only reason that an immune system would then attack this is because it has an antigen on it that's foreign. It's not recognized as the same. This could be for example, if you have a connective tissue thing here, cells, and then all of a sudden it's being attacked, something about it must have changed. There must be an antigen that's now not recognized as the same. The only thing that changes proteins and their structure, in this sense, would be a mutation. So sometimes random mutations can happen, which could lead to non-recognized could lead to non-recognized antigens. This could lead to, for example, e.g. cancer, right? And that gives an immune response to cancer. But as well as that, for example, in lupus or systemic lupus erythematosus or lupus or rheumatoid arthritis or allergies, you can have an autoimmune disease where the immune system attacks its own, attacks your own body because of something where it's now recognizing it as a problem. So for example, that could be because of a mutation changing alleles, or it could be the way that you inherit your antibodies, for example, or those kinds of things, like those alleles that code for them, just happen to go for that. So there's that, and also different antigens. So there's the genetic part. Now the environmental bit. Again, we're looking at how the environment shapes your specific immune response, your T and B cells. Well, first of all, you get illnesses, don't you? So you get exposure to pathogens. You'll have different different pathogens, You'll have different memory cells, won't you? So different memory cells. For example, those that have been exposed to chickenpox, right, will have memory cells to chickenpox. Whereas those that don't will not. So they will get it again, whereas a lot will be immune. As well as that, what else do we do to shape immune responses? I mean, we have them as kids. We have vaccines, right? That's going to produce primary and then, immune response and also memory cells as well. For example, your MMR vaccine, right? Allergies is another example. I have hay fever, for example, and I can't stand it. So, you have an immune response to pollen, and not everyone has that. What other things as well? For example, you could have HIV as well. So HIV infection can destroy those. When we're looking at genetics in the environment, our environment can influence the expression of genes through epigenetic changes as well. So you could have epigenetic changes due to, for example, poor diet or stress. If you're not eating and you're starved and you have barely any protein in your body, you're not going to be devoting that much protein to making hella antibodies, because you kind of need to have everything else in your body and not die. So there's that. So it's possible to manufacture antibodies to treat certain diseases. These are known as synthetic antibodies. So we have DNL-Fab3, and it's shown, and here's an example. To state two conclusions that can be drawn about the differences the way DNA-Fab3 functions and the functioning of normal antibodies. So you can see that there's antigen binding sites here, here, and here. Your normal antibody, right, is chilling, and it's got this, and there's your binding site there and there. So immediately, we have more than 2 antigen binding sites, right? So it means it can bind to more, well, actually antigens. Really, I just repeated that again, whoops. Now, as well as that, what else do we have going on? It doesn't have a constant region. No constant region. You need to explain what that means. Can't bind to immune cells. Constant regions are useful. For example, if you have opsonins, when you have an antibody acting as an opsonin means to make tasty for phagocytosis are eating cells, right? So if you have antibodies that are bound to a pathogen, and they have that constant region that makes it look tasty, as in tags it for the phagocyte, they can bind to phagocytes and cause them to actually phagocytose them. So here, it can't bind to immune cells, it can't do that. If you go a bit further as well, if I just draw a bit more detail here, we have this, and this, right? We have those disulfide bridges, we have a hinge region here and hinge region here. Now in this case, can you see how there's more hinge regions? There's hinging there, and hinging there, can we say? I think those are the main ones, to be honest. So all organisms exchange gases with their environment. Organisms can use simple diffusion to exchange gases when the diffusion pathway is less than 1 millimeter. So a beet armyworm larva has a cylindrical shape. It's 15 millimeters long, and it has a volume of 30 millimeters. It says calculate the diffusion pathway of the larva and state whether it could or could not rely on simple diffusion across its external surface to meet the gas exchange requirements, and use this formula: volume of a cylinder equals pi r squared L. Fine. So if we're looking for the diffusion distance, it's got to go in like this to the core, to the middle, right? To the center. It's got to go through here. So in order to do that, we need to find the distance from the outer part to the innermost bit here in the middle, because obviously we're not going to go all the way, it's just back out, right? So we need to find the radius. So they've told you that pi r squared L is equal to the volume. I'm going to rearrange this to get the radius. So radius squared equals V, the volume, over pi L. So then R equals the square root of a V over pi L. If I do that, we then get R equals the square root of, so the volume they've given it you is 30 millimeters cubed, so it's going to be 30 divided by pi times 15. If we do that, we get 0.787, which is roughly 0.8 millimeters, right? Can it or could it not rely on simple diffusion? The questions told you can if you're less than 1 millimeter in radius. So it's 0.8 millimeters because we want to do the same number of significant figures, and they give us 1, and it can because that's less than 1. There you go. So beet armyworm larvae eat a variety of plants, including tomato plants. Scientists wanted to investigate how effective a chemical called methyl jasmonate was in stopping these larvae from eating plants. They sprayed the tomato plants at different concentrations of these and recorded the final biomass. So independent is the concentration of this thing that they're using, and then the dependent is our final biomass. And then what's this question they're trying to answer? They're trying to see how effective a chemical is at stopping them from doing this. So they're using this to represent that, right? If we have a look at this data, then we can see that we have these bars that are equally distant from the mean, so those are your SDs, right? Your standard deviations. So we can see that as the concentration goes up, the general trend is that the final plant biomass is going up, which implies that they must be, eating less, right? Fine. So the student wrote this hypothesis: plants use methyl jasmonate as a defense against herbivory. What additional information would they need to confirm this hypothesis? The questions told you that they're spraying it on here, and they're just spraying it on different concentrations, and we don't even, know if they've used tomato plants, right? So first of all, if they want to do that, they need to know, do these actually produce the methyl jasmonate? So what do they need to know? They need to know whether tomato plants actually produce methyl jasmonate. The reason is that why do we need to know that? It's because we want to see, how much, they're making in comparison to the experimental amounts that they're using as well, right? So the whole reason is then we can see if they, kind of have them chill in, measure how much they make, and then give them these pests and see what happens. So, you would measure amount before herbivory, then see if this increases after, right? As well as that, this student says that they're saying plants use it against herbivory, right? So we also need to see if other insects are going in the same way, because our question was to look at how well this armyworm does it. This student is saying that it's insects or herbivores in general. As well as that, we don't actually know if the whole point of it is to kill it, basically. We just spot a pattern of having more of this means these are, being eaten less. It doesn't mean they're necessarily killing them or, stopping them like that. It might just, I don't know, make them like fly away and be like, oh, this stinks or something like that. We don't know. So as well as that, the effect on the actual population, are they dying? And also, do other insects respond in the same way? And as well as that, really, if they're saying plants in general, we've used tomatoes, do other plants do the same thing? So you'd have to do it on another species as well. So suggest one valid conclusion it's possible to draw from these results. So what that means is, what can you look at this and definitely say is the case? You cannot say, based on these results alone, a higher concentration means that these are being eaten less, because it doesn't show that. All it's showing is a higher concentration means the biomass goes up. That's it. So that's all you can conclude, is that increasing methyl jasmonate increases final biomass. That's all you can conclude from that data. You cannot say anything else. You can't just make things up that aren't shown there. It seems pedantic, but the reason is to make sure that every little itty bitty thing is tested. Otherwise, if we looked at this and, for example, I don't know, it was like amount of sugar eaten and then people's height, right? For some reason, and we saw it was a straight line like that. We were like, okay, and we were experimenting growth. If I then go ahead and say, right, if we eat more sugar, we grow more, right? That's a really bad conclusion to draw, because this, for example, only shows sugar and it's linked to height, which is confounded by like 8 trillion different other variables, like, lifestyle and other things that you're eating, drinking, and sleep and all that stuff, right? But also as well, it would mislead us into maybe shoving lots of sugar into things, and we know that's not a really good idea. So that's why we have to be really, really pedantic and say, look, if we look at these results, the only thing we can truly, really conclude is that high methyl jasmonate increases the biomass from that data alone. So they also recorded the level of cannibalism amongst these armyworm larvae. I don't know why I keep saying beetroot almost. Anyway, cannibalism was measured as the number of beet armyworm larvae eaten by other armyworm larvae. So they're looking at the proportion that were larvae eaten over larvae total, right? So here, a quarter of them were eaten at the total, right? And then so forth. So you can see that the concentration doesn't really change very much between these two. But then all of a sudden, between here and here, you can see there's more cannibalism. Only thing you can conclude from this is, for some reason, that using more than this gets you more cannibalism. Doesn't matter about anything else. It's literally just what you see here. So that means a higher than 0.1 millimole per decimeter cubed concentration of methyl jasmonate increases cannibalism. That's all you can conclude. There's literally nothing else you can get from that. So bacteria and fungi can be used to make food for human consumption. The use of microorganisms in food production creates fewer ethical uses and issues from the use of the animals. So using examples, describe and explain some other advantages of using microorganisms to produce food for human consumption. It's only advantages we're talking about. So what we can do is we can make mycoprotein, for example, or corn. That's something that we use. And in some microorganisms, you can make them actually use straight up like waste materials as well, which can, be then converted into useful foods as well. And that's good because you can get rid of waste products. As well as that, what other advantages? It means that you're able to grow them quite quickly and easily using a big fermenter, and they require a lot less in terms of their requirements to grow. Which means as a result, you're having to use less land, for example, growing like crops and livestock. You're getting less waste products out of this as well. And as well as that, you can also reuse them waste products in another thing if you can find a reaction as well. That works. As well as that, the proteins that are made tend to be more healthy rather than animal proteins, and as well as that can be suitable for things like, for people who like eat vegan or vegetarian food. So and those are a few of them. There's more as well that I'll get onto. I just want to yap too long. So one of them immediately is that like you can get less waste from this because the requirements of microorganisms for growth are simpler than like cattle, for example, that need to eat lots of grass and this and that and grain and whatever. As well as that, this is cheaper, and the waste can be used as a substrate from not only just these, if we can find anything, from other processes. Like, for example, molasses from sugar. You can use that. As well as that, this means that we can also it's also less damaging to the environment. So it's actually better for the environment. Why? It's because there's a couple of things. One, you need less land needed versus rearing cattle, less energy required because they grow at they can grow at lower temperatures. As well as that, we have less waste, right? Other advantages as well is it produces vegan food like corn, and as well as that, if we're living in England, we're not going to get really nice, like pretty, like swathes of fields and sun, right? So it means that it's unaffected by like climate, for example. So you can grow crops, well, rather than growing crops, sorry, I should say, you can get the materials from them from just, running a big fermenter. So it's unaffected by climate. Okay, and I've also forgotten, whoops, to write the explanations as well. So produces vegan food. Why? Because, it's, what's the word? There's fewer animal proteins, right, derived as well. And then in terms of unaffected by climate, why is that an advantage? It means you can produce in more, many locations. So you're not dependent on waiting for a specific season to have enough food. Then it says describe using examples. I'm just going to throw some examples in between. So for example, vegan food, corn. Other examples, for example, are yeast for bread. What else have we got? We've got fungal lactase for lactose-free milk. And then you would say an advantage as well. So for example, this means that people who are lactose intolerant can drink milk without having side effects and symptoms and all that jazz as well. So those are a couple examples. You would say the example, its advantage and why it's an advantage. On an industrial scale, microorganisms can be cultured using batch or continuous fermentation, and here's some statements about the industrial culturing of microorganisms. Place ticks in the table to indicate whether each statement applies to batch or continuous fermentation. So batch fermentation is where you grow microorganisms in a vessel, and when a culture's done, you take the whole thing out, you put new things back inside the vessel with the microorganisms and whatever, and you redo it. And this is also known as closed. Continuous is where you're just continually growing those microorganisms in a vessel without stopping. You're putting the nutrients in, you're taking the wastes out, and you're just constantly doing that while they chill in here. Whereas batches, again, in, take the whole thing out, in and out. So in that case, let's compare the two of them. So waste is removed during the fermentation process. Batch, the whole thing's being taken out, and it's not like continually going. You're taking everything out, and that's going to be with the waste in it as well and have to be separated separately, right? The waste is removed in batch after the fermentation process. Here, the key word is during. Continuous is while you're doing it while it's still fermenting, so that's continuous, but not here because you got to do it after you've taken it out. A fixed volume of nutrient medium is used. In batches, yes, because you put a batch in, you take the whole thing out, but with this, you're always adding more in because they're going to use that, nutrient while they're growing. So here, no. Secondary metabolites are more likely to be produced. Secondary metabolites are things that are made that don't directly contribute to an organism's growth, like they're not dependent to it, so they won't suffer without. So for example, if bacteria, like fungi for example, compete with bacteria, they make antibiotics. They don't have to, it doesn't directly contribute to them growing, it just kills off other stuff, right? So here, in continuous, we're continually removing all the garbage and rubbish we don't want. Whereas batches, again, we're kind of leaving in batches, we're going to go leave it there and take it and do its thing, and it's still going to carry on a bit here and there as well. And it's all mixed together with all the waste and other stuff too. And we will run out of like the right things as well, in nutrients, for example, in batch, whereas here you're constantly adding them in, so they may compete and make those secondary metabolites. So in that case, batch does, continuous doesn't. The growth rate tends to be faster. Here, batch, it will grow, right? And then it will drop because they'll run out of nutrients eventually, and then you have to start the whole thing again. Whereas here, it's continuous. You keep pumping them with what you need and taking the rubbish out, so it's continuous, not batch. And the culture is grown for a fixed period of time. That's batch, whereas these are continually growing down, growing down, growing down. So serial dilution questions, right? Follow the story and do it step by step. So serial dilution can be used to estimate the size of this. A scientist got a 20 centimeter cubed bacterial culture that had 10 to the 6 cells. So 10 to the 5, sorry. So here, 1 times 10 to the 6 cells was in 20 centimeters cubed, right? So if I do 1 times 10 to the 6 over 20, I can figure out how many cells you will have in a centimeter cubed, right? So that means we will have 5 times 10 to the 4 cells per centimeter cubed, right? So what they've done is they've diluted it by adding 1 of this and then taking 1 centimeter cubed of this and then adding 9 to make it 10. So they've done a 1 in 10 dilution, right? But then they've also done another 1 in 10 dilution as well, right? So if we do that, then we end up with 5 times 10 to the 4 divided by 10 and then divided by 10 again. So we end up with 500, right? Cells per centimeter cubed. Now, that's in our final thing. They've used 0.1 centimeters cubed of this. So if 1 centimeter cubed is 500 cells in it, 0.1 is going to have 500 over 10, which is 50. And there you go. So a student carried out different serial dilution to estimate the size of another bacterial population, and they resulted in 4 colonies developing on an agar plate. Explain why this estimation is likely to be inaccurate, right? Here, we've said, wherever it's gone, each colony developed is estimated to represent a single bacterial cell in the culture, right? So if we have a big plate, we've got 1, 2, 3, 4. That's pretty rubbish numbers, because if I do another repeat, maybe I got 1. Maybe I got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Which one's the right one? We don't know. So here you can see that the effect of the errors, we get random errors, right? That's going to be the case. It's going to be across all of these. So although, yes, 1 versus 4 versus, 1, 2, 3, 4, 5, right, isn't a huge problem in these cells, when we're scaling it back up, the impact is huge. For example, if I do a 1 in 1 million dilution, right? So wait, that's too many zeros, isn't it? 1, 2, 3, 4, 5, 6, yeah. So if I have a 1 in a million dilution, let's say I count 1 here, and then I count 1, 2, 3, 4 here, right? I'm assuming that this would have been 1 with 6 zeros, 1, 2, 3, 4, 5, 6. Whereas here, I'm assuming that's 1, if we undo the dilution, that is 1, 2, 3, 4, 5, 6. So you've got 4 million versus 3 million. There's a 3 million difference. Whereas if I count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, right? That's going to be 12, which then would be 12, 1, 2, 3, 4, 5, 6. And if I count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, for example, then that's 13, which is 13 million, right? Whereas if I do another one here, then maybe 1, 2, 3, 4, 5, and then maybe 0 there. Can you see how the scale that we end up going to, even though these look small on the surface, have a huge impact on the differences? Whereas if we count loads more, the impact of those small differences will produce less variation and errors, right? So that's what we're trying to get and turn into words. If you have low colony numbers with small differences, they will have large errors and more variation when scaling up. And that's exactly why. It's better to count loads and loads and loads of them, because then you'll have less variation. You'll have, instead of like, 1 and 2, you'll have like 1, 2, 3, 4, 5, 6, 7, 9, and 10. And then like, 9, 11, 15, 16, 17, 18, that kind of thing. Some microorganisms can be used in human industry. Some are pathogenic. Pathogenic microorganisms are transmitted in various ways. Complete the passage, right? So let's have a look. Some pathogens are carried between host organisms by animals, which are often insects. These animals suffer no symptoms of the disease and are known as. So they're carrying the disease and giving it to something else without suffering. It's not just carriers. You can't say that. It's got to be vectors. That's the specific word we're looking at for. Then it says other pathogens, such as P. infestans causing potato blight, produces reproductive structures called. So this is all to do with a fungus. It's a fungal disease, so it's spores. Anyways, thank you very much. I hope that helped. Please like, share, and subscribe, and you know the drill.